Today at Medical Sciences, we want to discuss about Rhesus blood group system. This Rhesus blood group system is the second significant blood group system. The first one was ABO, which we looked at the previous video. So in this session, we are going to look at another second clinically significant blood group system, which we call Rhesus or RH factor. And this Rhesus factor was first discovered in 1940 by the German scientist Karl Landsteiner. So Karl Landsteiner discovered together with Wayne. So these scientists, they are the one that discovered this Rhesus factor, and it was named so because it was discovered in the Rhesus monkey. It was discovered in the Rhesus monkey in 1940 by Karl Landsteiner and Wayne. So after its discovery, there are many theories that have been put forward to explain its inheritance, and according to Fisher and Rhesus, they have gone ahead to explain this Rhesus, whereby this Rhesus factor or blood group system is commanded by genes, and the most significant genes are 5, whereby we have genes that is C, we have D, small d, capital C, then middle C, or what we can call C, small c, then we have capital E and small e. So these are the 6 genes, but the most significant are 5. The 5 ones include D, capital C, small c or middle c, then E and small c. Whereas this D does not exist. Small d does not exist, so that's why we are saying we have 5 genes. So the presence of small d indicates absence of Rhesus D. And among these 5, that is gene D, capital C, capital E, small e, the most antigenic or the one which is immunologically active is gene D. So gene capital D is the one that is which is more antigenic, or it is immunogenic than both of them. So this one is the one which is clinically significant. This is clinically significant due to its much antigenicity. Why is it more antigenic? Because it has many receptors on the surface. So we're going to see that these genes, which were discovered by Fisher and Rhesus, they are inherited as a complex. First of all, they are inherited in 3 alleles, and these 3 alleles are located close to each other on the locus of the chromosome, on the loci. So these loci, they are closely together. They are always found close on the chromosome, and they are inherited as 3 alleles. Either it can be C, capital D, small e. So they are inherited as 3 alleles or as a complex. That is what we can talk about. And after inheriting these genes, they are the one that command formation of antigens on the surface of red cells. One thing I want you to remember as a student is that Rhesus D or Rhesus genes command formation of the antigens, which are only located on the surface of red cells. They are not found on leukocytes, they are not found on platelets, they are not found in other body fluids, only found on the red cells. So if we have the RBC, and maybe this is our red cell, this red cell can have different antigens has different antigens, can have different antigens. So we're going to see on the surface of RBCs, when an individual inherits a gene, especially D gene, that is responsible for Rhesus, which is the most antigenic, on the surface of red cell, this D gene commands the formation of D antigen on the surface of RBC. So this one becomes D antigen. And if an individual inherits C gene, it commands the formation of C antigen on the surface of red cell. So this one becomes the C gene. If an individual inherits middle C gene, that is small c, this is capital C, and this is small c gene, it also results into formation of small c antigen. If someone inherits capital E gene, it commands the formation of capital E antigen on the surface of red cells. So we're going to find that even small c, and this was capital C, this one is small c antigen, this is capital E antigen, and small e antigen. As we said that small d or middle d does not exist, its presence indicates absence of antigen D. So we do not have small d antigen on the surface of RBC. That's why we said there are 5. 1, 2, 3, 4, 5. There are only 5 antigens present on the surface of RBCs. And whenever an individual expresses these antigens on the surface of red cells, for Rhesus, we are going to see that they have no naturally occurring antibodies. So we're going to see that if an individual has antigen D on the surface of RBC, there is these ones, they don't have no antibody against it. So in short, with Rhesus grouping is different from ABO grouping, because in ABO, they have naturally occurring antibodies. But for Rhesus, they have no antibodies which are naturally occurring. They are not naturally occurring. They only come when there is sensitization. They only exist due to sensitization. And how does sensitization come about? Either after receiving a blood transfusion of a Rhesus negative individual with blood which is Rhesus positive. So if I have an individual who is Rhesus, a donor who is Rhesus positive, and I donate it to Rhesus negative, that one, the cells of this Rhesus negative individual become sensitized or immunized with antibodies against Rhesus. Another scenario is whereby a Rhesus negative mother gets a child from a father who is Rhesus positive, and of course automatically the fetus also becomes Rhesus positive, which goes and immunizes or sensitizes the RBCs of the mother. And the antibodies we are talking about in the ABO, they were of IgM type, but with Rhesus, majority of they are of IgG type, majorly IgG type 1 and type 3. And because they are of IgG type, they have ability to cross the placenta. So these ones can cross the placenta because they are of IgG type. And we have said they are not naturally occurring, but they are developed, or they are immunological. Why? Because they develop after sensitization or after being immunized with Rhesus positive cells. But we are talking about Rhesus positive and negative. We want to know how does it come about. We have seen different 5 antigens, including small antigen D. These 5 antigens, the most important, as we said, and more antigenic is Rhesus D. And we said Rhesus D is someone may acquire it or may not have it. So those, if this is an RBC, these are 2 different red cells. If these are red cells, this is an RBC and this is an RBC. If an individual has the D gene, capital D gene, is going to command formation of the D antigen on the surface of red cell. And this person with the D antigen on the surface of red cell is the one which is known as Rhesus positive. This one becomes Rhesus positive. That is the D positive because it's the one which is the most immunologically occurring. Whereas an individual who does not inherit D gene, if there is no D gene, which is always denoted by small d, meaning this person cannot express D antigen. So they have no D antigen. And absence of D antigen, it means this one becomes Rhesus negative. Meaning Rhesus positive, they have antigen on the surface, antigen D, whereas Rhesus negative, they lack antigen D because they don't have D gene to command the formation of that protein called the antigen. So this is for the sake of D, which is the most immunologically active. But with the exception of others, C, E, then capital E, small e, all these ones for them, they can also develop antibodies, except anti-small d. So these ones can develop, so we can find someone who can have, so under Rhesus we can have anti-D, we can have anti-capital C, anti-small c, we can have anti-capital E and small e, but not anti-small d because it does not exist. It only just indicates the absence. So these are antibodies, and we have said they are of IgG type, and they have ability to cross the placenta because IgG is very small. It is dimeric, and we have seen what an individual who is regarded as Rhesus positive, how they look like, and Rhesus negative. So the challenge with this, and which is we are applying in our hospitals during compatibility testing before transfusing someone with blood, we have to rule out is that individual positive or negative for Rhesus D, which is the most antigenic. So we can also see that the way we saw in this scenario, that if a person, I'm going to put it this side, so if an individual is Rhesus positive, can only donate, this person can donate to Rhesus positive patient. And if an individual is Rhesus negative, can donate to both Rhesus positive and can also donate to Rhesus negative. So this is according to the donation part. This is how they are compatible. But most importantly is that note, never give a Rhesus positive blood to the mothers of childbearing age, because if the mother is Rhesus negative and you infuse them with the Rhesus positive and they are of childbearing age, they are going to develop these IgG antibodies which have ability to cross the placenta and can cause or can react in case this mother got married to a man who is Rhesus positive. But this one is always solved by vaccination. You can always, in case we detect that the father is Rhesus positive, and the mother is negative, we normally give them anti-D vaccine. And this anti-D is normally given between 28 to 30 weeks of pregnancy. So it is always so. It does not stop you to, if you're a Rhesus positive man, to marry a Rhesus negative woman. Why? Because if it is known early during pregnancy, we normally give them a vaccine known as anti-D at the age of 28 to 30 weeks of pregnancy to prevent erythrohydrosis fetalis, or what we call hemolytic disease of the newborn. Because in case it is not given, we are going to see that this couple, if the man is Rhesus positive and the woman is Rhesus negative, upon fertilization, their child or the fetus is going to be Rhesus positive. And when it becomes Rhesus positive, because we said these are IgG type, they cross the placenta, this positive on the fetus, the mother's blood immune system is going to recognize it as a foreign antigen. And when it recognizes it as a foreign antigen, it is going to command the formation of anti-D antibodies in the blood, so that the subsequent pregnancies, these people by secondary immune response can come and destroy the fetus red cells. So if these anti-D are developed during subsequent pregnancies, that is the second, the third, and the fourth, they are able to cross placenta. And after crossing placenta, they destroy RBCs of the fetus, leading to a condition which we know as erythroblastosis fetalis. So this one leads to what we call erythroblastosis fetalis. That is destruction. So this baby can even die in the womb or after birth. And this is what can cause what we know as hemolytic disease of the newborn. You find a child is born with severe jaundice, and the cells are being destroyed at a faster rate, which leads to the jaundice. And that is what we call erythroblastosis fetalis, whereby the red cells are being destroyed in the mother's womb. And after being born, we are seeing this child developing hemolytic disease of the newborn, characterized by severe jaundice due to hyperbilirubinemia, because the red cells are being destroyed at a faster rate. So in summary, anti-D, the antigen or Rhesus D, is very immunologically significant because it can bring about what we call blood transfusion reactions leading to HDN. And to other individuals who are, you find a Rhesus negative person receiving subsequent transfusions of Rhesus positive, they also end up developing anti-D antibodies, which cause blood transfusion reactions and can even result into death if not regulated. And that's why we apply it every time we want to transfuse a patient. We must group the donor and the recipient to rule out these transfusion reactions so that we don't cause problem to our patient. So this is what we can discuss with you in this session. Thank you so much for always learning at Medical Sciences. Thank you so much.